This Week in Virology, the podcast about viruses, the kind that make you sick. This Week in Virology, episode number 82 for May 14th, 2010. Hey everybody, it's Vincent Racaniello, and this is TWiV, your weekly podcast all about viruses. And joining me today from North Central Florida is Rich Condit.
Hello, Vincent. How are you?
I'm well. How are you doing?
I'm great.
Having a great week?
Good time. Oh, yeah, having a great week. Very busy. A lot of balls in the air, but it's a good time.
No, as Dixon always says, busy is good.
Yeah, busy is good. That's right.
My course ended this week. I had a final exam.
Oh, it did?
And grades are due today, and we're still grading the course, the exam. So I haven't been in the lab much.
So did anybody learn anything?
I did. I don't know if that counts.
Yeah, no, that counts. I'll bet you they did too. That's good.
I think they did, and I told them I want them to go in the world knowing more virology than anybody else.
Yeah.
Or any other layperson, because that was my goal. So I think they did, and of course we'll get better next year. I learned some things, and we'll be even better every year. So, but now I'll have some time to do some experiments and put my head in the lab. So that'll be fun. So today we have just me and you again, because Alan Dove is doing family stuff, and Dixon, unfortunately, this is kind of sad. He's moving out of his office today.
What?
They're kicking him out of his office.
Wow.
It's kind of crummy. So Dixon, of course, is retired. He's a professor emeritus. He has a lovely office overlooking the Hudson River.
Is it near you, his office?
It's in another building across the street.
I see. Okay.
And they want to give it to someone else, so they're moving him. But here's the weird thing. They're going to give him another office, but it's not going to be ready till November.
Oh, great.
And so I think that's crappy.
Yeah, but you know what? Dixon will find a way to keep himself busy. He'll go somewhere, He'll go fishing.
Well, he told me he's going to stay home except for TWiV and TWiP. He'll be in here for those, so that's good.
Well, good.
I was afraid he wasn't going to come in anymore.
Oh, it'll be good to have him back on. It's been a while.
Maybe I'll get him in every week for TWiP too, and that twice a week I'll get him in here, and that'll be good. But anyway, I've it's kind of sad, but that's what happens to when you retire. It's the university isn't obligated to keep you on.
That's what happens when you die too. People just move right on by.
Yeah.
The world goes on without you.
Yeah, that's why you have to take care of you have to take care of yourself, right?
That's right.
So that's what a family's for too. They care about you.
Absolutely. Absolutely.
Well, today we have a couple of great stories as usual. Today we're back to the storytelling mode. And I don't know how long the first one is very interesting and requires a lot of background because it deals with immunology, and it may take us a long time, but we'll see what happens.
Okay. And
it is entitled, it's a Nature paper, Effects of Thymic Selection of the T-Cell Repertoire on HLA Class I Associated Control of HIV Infection. Now that is a mouthful.
Yeah.
That's 6 different things that we have to explain here.
Is this one of these papers that you read over breakfast?
I was actually reading this morning, yeah. It took me a long time to get through this one.
Oh, yeah.
Because
this is tough.
One thing we should tell our listeners is we're both virologists. But virology is a cross-disciplinary field, so to really understand it, you have to learn about cell biology, molecular biology, and immunology, as well as other things. And this is immunology, this paper.
Immunology is not my strong suit, but I'm learning.
Exactly. I like it.
We have a journal club, an immunology and microbiology journal club that all of our students attend and stuff. And I pick up a lot of immunology there. And I lean on the graduate students to to answer my questions, and they help a lot.
Well, this is my weakest area, I think, immunology, but I'm learning as well. And in fact, our department is now microbiology and immunology. Mm-hmm. They're hiring immunologists. Right. So this morning I went into one of their offices and I said, hey, explain this business to me. So
Okay, well, explain it to me.
So this is about HIV infection. So let's do that first. When you get infected with HIV, virus enters you, starts multiplying in your T cells. And there's about a 2-week period where it looks like any other acute viral infection. You have a burst of virus in the blood. You have fever, malaise, achiness. Could be almost any viral infection. And then the amount of virus starts to drop. Your immune system kicks in, starts to drop. And then you have a period of time where there are low levels of virus, but it's still there. And this can go on for 10 years, and eventually you die of immunodeficiency. The virus destroys your immune system, and you get other infections.
And one of the main reasons it continues on for so long rather than being snuffed out completely is that it is integrated in a permanent state in the infected T cells.
That's right. The DNA's there. You can't get rid of it. The virus varies. It escapes every immune response you throw at it. Antibodies, it escapes. T cells, it escapes. And eventually you lose.
Drugs, it mutates again, acquires drug resistance.
Right. Now, we can control it with 3 drugs at once. That's pretty good.
Mm-hmm.
Triple therapy, but not everybody can afford that. Now, that is a course of infection, a typical course, but there are actually two other different kinds of courses of infection. This was actually a question on my final exam. What are the 3 different courses of HIV infection? So one is this typical progressor, it's called. You have the acute infection, 10 years, then you die of immunodeficiency. You have a rapid progressor, where you have, again, that initial acute phase, and then in a year or 2, you're dead. Everything happens really quickly. And then you have what are called elite controllers. And these people have low levels of virus, very low, forever. And some of them live fine. They never die. They never die of immunodeficiency. And these are pretty rare individuals. And we've actually talked about them before here on TWiV.
Right.
Obviously, it's interesting to know why they're so able to control infection, right?
Absolutely. You might ultimately be able to take advantage of that.
Yeah. So it's known that these individuals have a really good ability to clear or keep virus low as a consequence of their immune response. Now, when you're infected, 2 kinds of immunity kick in. Antibodies are made, and T cells. The T cells are what the story is about today. The T cells their job is to kill cells that are infected with virus. And a story that we did last year, I think, on TWiV, showed that T cells from these elite controllers are really good at lysing or killing HIV.
Yeah, I was trying to remember that. They're really good killers, right?
Exactly, yeah.
A granzyme thing.
Really good killers, something to do with granzyme, which is a protein that they put into the target cell to help break it open. All right, so that's elite controllers. They have a good T cell response. And in this paper, what they say right on is that elite controllers have very specific HLA class 1 alleles. So now we have to explain HLA. Human leukocyte antigens, or major histocompatibility complex. Incredibly important cell surface proteins for immunity. All right, and so in terms of a T cell that's looking to kill a virus-infected cell, the way that T cell identifies that this cell is virus-infected is that on the surface of that cell there's a HLA molecule, HLA class 1, and it's holding in its hand a little piece of the protein from the virus. So inside the cell, the virus is growing, it's making proteins. The cell is chopping up those proteins, and makes peptides out of them, puts them up to the surface and puts them in the MHC or the HLA molecule. And then
which are the same thing. 2 different names for the same thing.
Right. And the T cell then will recognize it and go, this is a virus-infected cell, kills it. And the important thing is not only do you have to have this virus peptide there, but it has to be displayed in this HLA molecule. Right. Absolutely essential. And that's kind of a quality control to make sure you don't go killing everything. Okay? So far, are we okay?
Yep, so far we're okay.
So these HLA molecules in humans are polymorphic. That means there are a lot of them. And there are something like 5 different loci of genes in humans and over 100 alleles of this HLA1. And it turns out that the variation of these genes in the human population is really important for controlling infectious diseases. So in populations where there's not a lot of variation in these HLA molecules, like island populations, they tend to be more susceptible to infectious diseases. And on the other hand, when you have a lot of interbreeding or outbreeding, I guess, you have a more diverse HLA display in your population, and they can better control infections. So the bottom line here is that these are very variable proteins.
And important, variability can be important in controlling infections.
I guess you could just, without even knowing anything, that would make sense, because the ability to display different peptides or interact with different T cells is conferred by this variability. Right. So that's important. So there's one allele of the HLA class 1 called HLA-B57. It happens to be a specific protein sequence. And in people who are elite controllers, there's a lot of those. So this has been known for some time now to be involved in some way in helping these elite controllers not die of AIDS. All right, so in this paper, they're trying to understand that. So remember, HLA-B57 or any HLA is going to present a peptide from the virus-infected cell to a T cell, and then the T cell is going to kill it. And what we know is that these HLA-B57s, for some reason, are better at that than some other HLA type. Right?
Right.
That's actually the background. Now, we actually need to know a little bit more about how T cells actually develop.
This is what I really learned from this, because this is the first time I really had to think about this. Okay.
Yes.
And this is subtle.
So T cells are lymphocytes, of course. They originate in your bone marrow, but to mature and to be ready to do what we've just been talking about, they go to your thymus, which is a gland that we all have, or most of us have. If you don't have one, you have a lot of problems with infections. But there are people without thymuses. And the T cells go there early in life, and they mature. And what does maturing mean? So the molecule on the surface of the T cell that engages this HLA with a viral peptide, it's called the T cell receptor. And there are a bunch of genes encoding this T cell receptor in the T cells. And as the T cells are produced in the thymus, these T cell receptor genes begin to rearrange and recombine. The result is every T cell that's made has a slightly different T cell receptor on its surface.
Kind of like an antibody, so that the variety in the T cell receptors will cover almost any epitope, any binding partner that you can think of.
Exactly. So you get all these millions and millions of different kinds of T cells produced. But then what happens to them is they go through a kind of selection. So what do we need for these T cells? Well, they have to be able to bind peptides presented by HLA. If they don't bind well, we don't want them. But if they bind too well, we don't want them either. So what kind of peptides are we talking about? It turns out that in the thymus, these T cells are shown host-derived peptides in the HLA molecule. So within the thymus, peptides, host peptides, our peptides are chopped up, they're put in HLA molecules, and then the T cells interact with them. And the T cells that don't interact are thrown away. They're ignored. The immunology texts say they're ignored, and they die of apoptosis.
Right.
Then the ones that bind too strongly, those are probably going to cause autoimmune problems. So they're also ignored, and they die. And the ones in between are kept. So those kind of bind moderately well to these host-derived peptides. Is that your understanding?
That's my understanding, but now I have a question. Are they are at the same time learning to differentiate between self and other. Is that correct? Because they're being shown self-peptides.
No, see, that's the thing. is key here. They're only being shown self, right?
Right.
So the implication is if you bind really strongly to self, that's all you're ever going to bind.
And that's a problem.
And that's a problem. So we want to get rid of them.
Okay.
The ones that are kind of fair to middling in terms of binding, those are kept.
Okay.
And of course, the ones that don't bind at all are thrown away.
I get it. Okay, that's great.
So actually, this is referred to by immunologists as positive and negative selection. So we're positively selecting for T cells that can bind peptide HLA, and then we negatively select for the ones that bind too well.
Right.
It turns out that, I don't know, about 10 to 15% of all the T cells that you ever make in the thymus actually get out. So it's not a lot. The rest are killed.
Right.
Because they don't suit those criteria. And so that's how they're selected. Then they leave the thymus, they go to your lymph glands, and then they just sit there and circulate and look for bad things like viral infections.
Right.
And then when one of them encounters an antigen, a viral antigen, they're always sampling the cell surface, looking at the HLAs and saying, is there something unusual here? And if they detect a viral antigen, they begin to proliferate, and you get tons and tons of the same T cell specific for that epitope. Now, the thing about HIV, this happens in an HIV infection, but then the virus changes its protein sequence, so the peptides aren't recognized very well by the T cells.
Right. You get infected with one virus to start with, and by the time you're done, you have a whole just bunch of different types of virus. The mutation rate is so high, and there's so much variability. It's amazing.
Right. So now the basis for this paper, which, by the way, is entirely computational, almost all computational. The idea is that, so we've already talked about these T cells are given peptides to test them out, right? Host-derived peptides. And the theory is that the diversity of these peptides changes what kind of T cell receptors are going to develop. And they cite an experiment here, which is very interesting. If you make mice and give them only one peptide in the thymus, not all of them. So they're instructing their T cells now with just one peptide. You can do that by experimentally manipulating the mice. They make T cells that are highly cross-reactive to many different peptide variants. Okay, so you have maybe, I don't know what the length of this peptide is offhand. Do you?
Like 9 amino acids or something like that.
So, you change a couple of amino acids in this peptide. And these T cells derived from these mice that have only developed with one peptide, they can handle that pretty well. But if you give them a lot of peptides early on, the T cells aren't as good at handling changes.
Right. Okay.
So that was one of the previous data for this paper. So again, remembering the context of HIV infection, it's always changing these peptide sequences. So if the T cells can't handle that, the infection will continue. So somehow the elite controllers can handle all the variants that arise. So in this paper, they use algorithms to predict whether a peptide will bind to a given HLA molecule. Okay, and they did this in silico experiment, first of all, where they take all the possible peptides derived from human proteins, which is called the human proteome, right? So you can computationally, figure out all the different peptides you could derive. It's a lot. So I think it's 10 to the 7th.
Yeah, 10 to the 7th.
And then they say, what will these peptides bind to?
And this must be based on structural modeling of these HLA molecules.
Yeah, I think that's correct. And a lot of math.
A lot of math. There is a lot of math.
Yeah, because this is 10 to the 7th sequences, and then you have to fit each one in different ways. It's computationally intensive, yeah.
Mm-hmm.
So what they say of these 10 to the 7th sequences, only 70,000 are predicted to bind to HLA B57. Okay, that's the protective HLA allele.
Right.
Whereas 180,000 will bind to another allele that is not protective against HIV. So this is interesting because you would think that binding more peptides would be good, but apparently it's not. It doesn't correlate with being protective. That's the first clue that comes out of this paper, that more is not better. All right? Everybody okay so far out there? You might want to stop and rewind.
I'm okay. There's probably some people rewinding, but that's okay.
Yeah, it's
I'm all right.
We thought we'd really...
Of course, I cheated. I read the paper.
Yeah, I mean, hearing it is probably tough, but we're trying to...
Yeah, you're doing okay.
Go through it a little slower. All right, so the idea is that... their idea is that in the thymus, if you give the T cells fewer peptides, maybe then the T cells that are selected will be able to handle more different peptides when they encounter them later on.
Based on that critical experiment that you cited early on, where if you give the mice just one peptide, you wind up with T cells that can handle lots of different peptides.
Right. So you're agreeing with that setup? Now that's all correct, right?
Absolutely.
So that's their hypothesis, and then they tested it in silico, as they say. So they take a number of peptides and T cell receptors, and they know the structure of those, and they try and predict which is going to interact with what. And they use computational models, string models, they call them. I suppose they involve string theory, right?
I guess.
But I don't know. I don't quite understand that. But they can just determine what is going to react with what, okay? So then they say, given certain peptides that we add, we can tell which of these T cells would come out of the thymus because they're above, they're binding, but not too much. So they can predict the strength of the binding and say which peptides would select what kind of T cell receptors, right?
Right.
So what they say is that the calculations predict that the T cells that are made or instructed by B57, HLA B57, okay, these are presenting fewer self-peptides in the thymus. These can recognize peptides from viruses that have changes in them because those peptides, the original peptides that instructed those T cells, had just a few important contacts between the peptide and the HLA molecule. In contrast, the T cells that are restricted or that interacted with the different HLA molecule, they're selected but with peptides that have a lot of different contacts to interact with the HLA molecule. And if you change any of those, you screw up the binding. And so those are less likely to deal with viral changes because there's so many contacts required for the interaction of the peptide with the HLA that just one change screws it up.
I think of this as kind of in the thymus, these HLA presenting cells are educating the T cell population.
Right.
And the idea is that this HLA B57, that there is a correlation between not binding a lot of self-peptides and winding up with a very diverse, a flexible, put it that way, flexible T cell repertoire that can handle changes in peptide sequence, the type of which you might see as the virus is variable, as when the virus goes through this variation. So in this education process, this HLA B57 educates a T cell population that is a lot more flexible in terms of its binding to virus-derived peptides. There can be variations in peptide sequence, and the T cells can still handle them. Whereas T cells educated by other HLA types that bind during this process, this go-to-school process, bind more self-peptides, are much less flexible. So the virus can go through variation and get around those guys.
Right. And
very interesting.
The key seems to be how you educate them. If you give them a lot of host peptides to educate them, it's not good.
Right.
Because then they end up binding the peptides with a lot of interactions between the peptide and the HLA, which could be easily disrupted.
I don't quite understand why that should be, I guess. Do you?
No, that I do not understand.
I'm not sure they understand it either.
I thought about that a lot. Why giving you a diverse peptide repertoire does that? I'm not sure. No.
Maybe we'll get an immunologist that'll write in and tell us.
But intuitively, if you have just like two contacts between a peptide and the HLA, and you can see that there is a lot more room for changes and not affecting the interaction, right?
Right.
So
right.
2 out of 9, you still have 7 places you could change because they're not
Right.
contact places. But if 6 out of 9 are contacting, you change any one of those, you mess it up. So that makes sense to me, right? But I don't know why the diversity makes that big a difference. I tried to see it in here, but I didn't get it. So that's basically this. This is explaining in a computational way why B57 might be more able to, well, it shows that B57 can handle different sequences because they modeled that computationally. And it may be as a consequence of this education.
So we mentioned last week, we talk occasionally about this concept of virus equilibrium and stuff. And last week we had a discussion about how maybe HIV may ultimately evolve to coexist with humans. And we talked about the possibility that some of the ape immunodeficiency viruses that have been with apes for a much longer time don't seem to cause disease. It would be really interesting to look at those situations and look at this relationship between their HLA type and the virus, and see if what's been learned from this, or some of the ideas that come out from this, apply to that situation. Yeah. I mean, you could see how actually this got me thinking about how reading this paper got me thinking about how modern medicine tinkers with evolution. Okay, because you can see how in a completely natural situation, this winds up selecting for people who have this particular HLA type because they can control the virus infection. They don't die, and it influences the pathogen, would ultimately, I would think, to some extent, influence the evolution of the host. Of course, we intervene with this, and so that's not going to happen in the same way. But it would be interesting to look at other creatures, apes, that have learned to coexist with these viruses and do the appropriate immunology with them. I'm not sure you have the tools to do it. But see if some of the principles that are highlighted in this apply to those situations.
No, it's a good point, because the primates have been living with the virus for much longer than we have. Mm-hmm. So there's been plenty of time to select the right HLAs. So I guess you need to have the structure of these proteins and know what kind of peptides. And you could do a similar experiment as was done here.
Right.
As far as your point in medicine and selection is very well taken, because if we give people drugs, you never give us a chance to select or be selected, right? Because the whole idea is you kill off the people who can't respond well, and then the ones that do survive and their offspring predominates. But it takes a long time. And if you, I've thought about this in the context of many infections. If we intervene, you don't allow for that with either vaccines or antivirals, right?
That's right. So, but, take your choice. What do you want? The tendency is to intervene because people don't want to be sick. People don't want to die. So we do that. But there is a consequence that we have to think about.
So this is interesting in the context of HLA. I was reading a little bit about it, and they said about 150,000 years ago there was a constriction in the HLA diversity.
Yes, I saw that, and I didn't. Do you know what that is?
I don't know what that is, but it's interesting that it was more diverse, and then maybe that was bad for it to be more diverse. And then
I remember this coming up in a previous show about how the variation in HLA among humans is actually narrower than a lot of other creatures, and it must have to do with this particular bottleneck.
Right. And we should say that whenever you have an infectious disease and you have people who are more susceptible than others, one of the first things that's looked at is the HLA type, right, to see if there's some correlation. And then if there isn't a correlation, then you move on to something else. But this is a really important determinant. I guess the last thing we should point out is that they did try and validate these predictions in human studies. So they had two populations of HIV patients. They're called cohorts. And we know the HLA type of these individuals. So they have 1,110 elite controllers and 628 progressors who have a lot of virus. And they can look at their alleles, and they actually did peptide binding studies on just a few of these individuals. And basically having HLA that binds more peptides is bad. They're going to be, those are the progressors.
Or self-peptides.
Self-peptides, that's right. They're looking at self-peptides. And the HLA, which is B57, and there's a second one called 27, they bind a lower fraction of self-peptides. So it's consistent with their prediction. I guess there are a lot of other things you can do now to test this.
Right.
Especially you could do it in an animal if you had the data we were just talking about. Okay?
Great.
I hope everyone got that. It's really, really interesting.
Just keep rewinding. You'll get it. Rewind, rewind, rewind.
It's good stuff. Yeah. All right. Okay, let's move on to our second story. But before we do that, a word from Data Robotics, the makers of Drobo. And Drobo, of course, is a storage solution for your computer. A wonderful box with multiple bays for hard drives. They have a pretty big product line now. They started out with one, but now they have 5 different products with different performances, connections, interfaces, numbers of drive bays. Maybe there are too many for you to decide what you want. Let me tell you what happened to me. I started out with a Drobo, the 4-bay original system. I wanted to protect my photographs, my videos, and music, and I filled it up. Eventually filled up, and even in that time, I used it for a couple of years. I had a couple of drives fail. So a red light comes on in the front. It said, oh, this drive has failed. I pop a new one in. The data is all restored. I don't lose anything. I can't tell you how important that is. You don't really have to think about it. You don't have to back up. You don't have to swap hard drives. You just put it on that, and it works. Eventually, I had too much data. It wouldn't fit on a single Drobo. I bought a second one that was a second generation, 4 bays, but had a FireWire interface. I had one of those at home. Now I have one here at school, and it just gives me more storage, and it's faster. Of course, doing podcasts, I make a lot of data. I videotape all my courses. Videotape. There's no more videotape. I record. I don't know what I guess we just say video record all of my lectures, and I don't want to lose any of that. I put it all on my Drobo. And I feel safe that it's not going to have a problem because, again, if anything fails, I can swap out the drive. And now I have a Drobo S, which I recently got. It's a little faster interface. I can actually do editing right on that unit. And that's a 5-drive unit, which has USB FireWire and eSATA connections. Really great. I have to emphasize to everyone, if you're really looking for simple, safe storage, you really can't beat this solution. Now, Data Robotics recently came out with a new model called the Drobo FS. FS stands for File Server or File Sharing. It's designed to share data or back up a small network, like up to 32 computers. You can hook it right into your Ether network. So if you need a storage solution that works on a network, the Drobo FS is for you. And again, it's very simple, very secure. Now, we're giving away a Drobo S. It's the third one. And we've given away 2 so far, of course. The third contest is really simple. Just go to bit.ly/TWiVDrobo, or you can go to twiv.tv/Drobo-contest. The rules are there. But it's basically a questionnaire, a 19-question questionnaire. It has to do with what kind of people listen to podcasts and whether Drobo would be interested in supporting podcasts like they do support us. So it's kind of an important questionnaire for you to take because we'd like to have their continued support, but if podcasters don't appeal to them, then it's not going to happen. So 19 questions, all multiple choice, no essays, no fill-in.
Piece of cake. Take you 5 minutes.
go over and do that, okay? And at the end of the 3-week period, they'll randomly pick out one of the respondents, and you could get a Drobo S for free. So how
Good
can
deal.
you beat that? And if you don't win, go to drobo.com/TWiV. They have made a page just for TWiV listeners, and you can use the code TWIVPOD to get $75 to $500 off a brand new Drobo. And of course, we thank Data Robotics for their support of science education this week in virology. It's good stuff, right?
Great. Yep. Absolutely.
Okay, our second paper today is another cool one. And this has to do with dengue.
I'm really glad you found this one because I've been aware of this issue for some time and never had a really very good grip on the why. And this not only helps me understand why, but it's an interesting paper itself.
Yeah, it's a good teaching tool because we always teach dengue, as you'll see why, and we can never give any support for the theory of why dengue is a serious disease, and this is really the first experimental support. So dengue is an illness, a viral illness caused by a flavivirus. It's an RNA virus with an envelope. It's transmitted by mosquitoes. So it's in many, many parts of the world. It's in many tropical regions, but it's also spreading elsewhere. As the used tire trade brings the mosquitoes around the world, so it brings dengue virus. Now, there are 4 serotypes of the virus. They don't cross-react to the extent that if you had immunity to one, you would be protected from the other. You get your first dengue infection, you get, kind of a nasty illness, but self-limiting. You typically don't die.
It can even be asymptomatic, I think.
Sure. Can be asymptomatic. You can have fever. You can have joint pain, but you get over it, and you have antibodies to whatever serotype you get infected. But it can also cause this potentially lethal dengue, hemorrhagic fever, and shock syndrome. And that almost always happens in people who have had a previous infection.
With a different serotype.
It's always a different serotype, right. So over the years, the theory has arisen, well, what is happening here? Let's say you get infected initially with serotype 1, and then you get a second infection with serotype 3. You get a memory response upon the second infection, so you make antibodies to serotype 1, because there's enough cross-reactivity to do that. Those serotype 1 antibodies will bind the serotype 3 virus, but they don't neutralize infectivity. What they do is allow that complex, the antibody-virus complex, to attach to a cell that has a receptor for the antibody. It's called an Fc receptor. Gets taken up into the cell, and then bad things happen. And the idea was this happens in macrophages, which are not usually infected by dengue, and then that's bad. They release cytokines, and that's what gives you the hemorrhagic fever and shock syndrome.
So one of the issues here is that this idea of serotypes and cross-reactivity is not absolutely perfect. If you mount an immune response to one virus, that antibody won't neutralize the other virus infection, so you can get infected with it, but it still binds a little bit, okay? And that turns out to be bad.
Right. And the key point is that it lets the virus into cells where it wouldn't normally go.
Right. It actually helps those viruses bind, that binding of that antibody to the virus helps those viruses bind to cells that it ordinarily wouldn't infect.
Right. So just to give you an idea of the numbers, 2.5 billion people in tropical and subtropical regions are at risk for infection, 50 to 100 million cases annually, 500,000 severe, and 15,000 deaths. So not on the top of the infectious disease list, but certainly serious, and there's a lot of work trying to figure out how to get around this.
I also like the other name for this is breakbone fever.
That's right.
Because when you're sick, some of the symptoms just feels like somebody's gone through and crushed all your bones, especially
That's right.
It's very painful.
Yeah, exactly. Okay, so now in this paper, the cool thing here, there's never been any evidence for this hypothesis. By the way, when you put an antibody on a virus and it gets into a cell by Fc receptors, that's called antibody-dependent enhancement of infection, ADE. Okay? There's no evidence for it, just theoretical. So here, for the first time, they have a mouse model that basically does something that looks a lot like antibody-dependent enhancement, and you get a similar lethal disease. So basically what they do is they take mice. There are a couple of caveats, but first let's describe the disease. They take mice, they infect them with dengue virus, either with or without antibodies to the virus, which have been raised in another mouse by infecting them. Right? Simple experiment. Guys that don't get the antibody, they're pretty good for a while. Eventually they die, they have paralysis 7 to 12 days later, which is weird because I don't think that happens in people, but it's a mouse model. The other guys who got antibody along with the virus, they have severe disease within 4 or 5 days.
Right, so you can clearly discriminate between those guys and the other guys. They get a severe illness. And in that way, it models the human situation, where if you ordinarily get dengue, you get a disease, but if you get dengue hemorrhagic fever because you've been previously infected, it's a much, much more severe disease.
Right. So is this like human? Well, humans make a lot of cytokines in this dengue hemorrhagic shock syndrome. So they look in the mice and they find elevated cytokines of a variety of kinds. So that's sort of a similar thing. First thing. Where does the virus replicate? Well, in these mice that get the antibody, it turns out the liver is like the primary place where the virus first goes. And then it spreads elsewhere, and that's distinct from virus in mice without antibody. So the liver seems to be a target. And the Fc receptor is important, so they can repeat this experiment, virus plus antibody, and then they add a second antibody which blocks the Fc receptor, and you prevent this severe disease. So that tells you that Fc-mediated uptake is involved, right? That's important. Right. And then they find that it's actually the endothelial cells in the liver that are being infected by the virus. And so that's interesting because part of the syndrome is a hemorrhagic syndrome in people.
Right, so that all makes sense.
Your little capillaries are breaking and bleeding. So this is consistent. The virus is replicating in endothelial cells. So there are lots of parallels. It's really the first good model for this syndrome.
And the other thing they do, I'm trying to find where the data are in the paper, and I can't quite come up with it, but correct me if I'm wrong, but they do an antibody titration, right? Where they put different amounts of antibody in these animals, because the theory is that if you put enough of even a crummy antibody, you should be able to deal with the infection. And the problem is having just the right, or if you like, the wrong amount of antibody so that you can bind some of the virus but not neutralize it. And they do titrations to verify that hypothesis.
Right, that's right. So these are low levels that do this. So
the one trick about the mice, I mean, you would think, okay, so why didn't somebody do this before? The trick is that the mice are interferon receptor knockouts.
Yeah.
Okay, so if you try and do this with a normal mice, the interferon squashes the infection right away, and you can't see any of this. But if you use interferon receptor deficient mice, then the interferon's out of the way, and you can see all this other stuff happen. And one of the interesting points that they make is that the virus ordinarily has an interferon countermeasure in humans, I believe.
That's right, but not in mice.
but not in mice. Yeah, that was it.
That's why you have to use these interferon receptor knockouts.
Exactly.
Yeah.
So that all makes sense.
Yeah, I mean, if you put this virus in normal mice, nothing happens, and so it's a little odd. And they do admit it in the discussion. They said this could be an odd thing, but it gives you some clues at least. I mean, they make this point that, look, the antibody lets the virus get into cells that normally wouldn't be infected, and that's what we think is happening in people.
They covered a lot of caveats in this discussion. And there was this cover story in the same issue, a preview that went along with it that covers a lot of the same caveats. It's almost like the guy who wrote the preview reviewed the paper.
Yeah,
and then the authors covered all of the stuff in rewriting the paper in their discussion.
Yeah,
but that's fine.
I suspect they didn't have a lot of these caveats initially in the discussion.
Yeah, but that's fine. That's
okay.
the way the process ought to work.
It's fine. I mean, it's getting published. It's fine.
Yeah.
The other thing that's interesting here is that this is a mouse-adapted virus.
Yes, that's right.
It was passed 10 times in mice, between
that's
mice
the
and
other
mosquito
trick to make
cells.
this work. They needed a mouse-adapted virus.
Right, and apparently there are a couple of amino acid changes in the viral glycoprotein that allow this to happen. So that's important too. So there are two things they mentioned that too. This could be weird. It could be a weird property, but it's fine. Animal models are never perfect, so all they do is give you clues, and that's what this is going to do.
This seems awfully close. I mean, it's they've reproduced a lot of the phenomena pretty closely, so it looks pretty good to me.
No, I think it's important to know that this can happen. It's not just a theory, because we didn't have much evidence in people.
So the other thing is that there's another, if I've got this right, another practical outcome of this, and that is that we've talked about this before, the original respiratory syncytial virus vaccine, did it not cause an exacerbated disease, and so they withdrew it, and the thinking was that it was a similar type of thing? Have I got that right?
That was one of the theories, right?
Where it was basically through a similar mechanism causing the same thing. The vaccine was giving you an immune response that probably wasn't quite good enough, and so that when you got infected, it actually exacerbated disease in some people, rather than clearing it up.
Yeah, so now, I mean, since then, it's been shown that one of the problems with that vaccine is that the inactivation process, the formalin, destroyed the virus so that it was no longer a good trigger of innate immunity.
I see, okay.
And so the antibody response was very, very low affinity as a consequence, because you need to have a really good innate response to get good antibodies produced.
Okay.
So, that explained why it wasn't very protective. But the atypical disease, I'm not sure we understand that still.
Okay.
Yeah, so I think what you said is correct. There is that theory still out there that somehow these antibodies led to inappropriate infection, maybe because they're low affinity and they didn't block infection but enhanced it.
It sure smells the same to me.
Could be it, yeah.
So my point is that this is not necessarily just a phenomenon that's peculiar to dengue. But this idea of antibody enhancement of infection is something that could have practical applications in other virus diseases and also in vaccinology.
Absolutely. Yep. So we'll learn a lot from this. I think it's great.
Yeah, it's nice to have the model. That's the most important bottom line from this. Well, two important bottom lines. One is that it really locks down this particular theory, and two, it provides the animal model to continue to do these kinds of experiments.
Yep. So that's actually been out for a while. I've been meaning to talk about it, and so there you go. Mouse model of severe dengue disease. I think we should move on to email. What do you say?
Sure.
We have a couple good ones. So the first one is from Eric, and it's actually an audio file. So let me see if I can get this to work here. I'll play this in a way that Rich can hear.
Hey, Dr. Racaniello. My name is Eric, and I'm a pathobiology major at the University of Connecticut. I recently watched a TED Talk on YouTube in which Kerry Mullis, the inventor of PCR, outlined his work on an entirely new form of antibiotic, which he says so far has been successful at ridding mice of anthrax. Basically, the antibiotic utilizes a pre-existing immunity that all, or at least most, humans have to something called alpha-gal epitope. This molecule can be attached to a DNA aptamer that preferentially binds to a particular protein on the surface of an invasive bacterium. This way you can immediately tap into an immune system that's already there, and you don't have to wait 5 or 6 days to develop. I think this idea is really clever, and I was wondering if you think this would also work as an antiviral treatment. For example, if the DNA aptamer that the alpha-gal epitope is bound to were to preferentially bind to the active, highly conserved site of GP120, which is the glycoprotein that HIV uses to enter T cells, could that possibly be used as a treatment? I figure if the virus mutated so that the DNA aptamer wasn't able to bind to GP120, then that would probably mean that GP120 also wouldn't be able to bind to the CD4 receptors on T cells, because its active site would have to be altered. So let me know what you think, and I love the podcast. Thanks, guys.
All right, thanks, Eric.
Great. Yep.
Did you get that?
Yeah, I got that. I got that.
A little muddled, but I think you could get it. So I went to YouTube. I found this video. Kerry Mullis at a TED Talk.
Kerry Mullis is a character.
He is. I sat next to him once at a dinner. He's quite a character.
Uh-huh.
So we have all this antibody called natural antibody. It's against alpha-galactose.
So explain to me why we have this again. It's because of it's to bacteria in our guts or whatever?
Right, so this disaccharide galactose, alpha-1,3 galactose, okay? We don't have this. We don't have this. We humans, apes, old world monkeys don't have the gene to make this 1,3 linkage between galactose.
Right, so it is not self.
It's not
That's
self.
important.
And the bacteria in our gut have it.
Okay.
Those are good bacteria. We like them. They like us. And we make antibodies against this alpha-gal because it's not self, right?
Okay.
So we have about 2% of our antibodies in our body, in our blood, are directed against this alpha-1,3 galactose. And it's called natural antibody because it's thought to be sort of like an innate response. You know, you don't need any priming. You don't need an adaptive response. It's there.
Right.
So in an early stage of infection, this antibody plus complement could lyse, say an envelope virus. So influenza virus, if it were produced in a animal that has alpha-1,3 galactose, it would be neutralized. Or any other virus, an envelope virus that were produced in an animal that infected us should be destroyed by these antibodies. Now, of course, it doesn't do anything for viruses that come from another person because they don't have alpha-1,3 galactose. Okay, so this is just maybe to prevent, most zoonotic infections.
Right.
But parenthetically, Rich, this is why we heat serum.
Yeah, I was thinking about that because I knew that heating serum was to inactivate complement. Tell you the honest truth, I usually don't pay any attention to it. It doesn't cause me any problems. But
yeah, if you grow your virus in human cells, there's not going to be any alpha-gal, so it's not a problem. But if you happen to grow them in animal cells, you should heat and activate your serum.
All right.
And so you don't get... I don't do it either for polio because we don't have any glycoproteins in it anyway.
Right.
But you guys do. Your viruses are glycosylated, so...
Yeah, well, probably everything I've done for the last 30 years is wrong.
You don't want to find out, do you?
No. No. I'm not changing now.
So that's alpha-gal antibody. Now, what does Kerry Mullis have to do with it? Okay, we have 2% of our antibodies of this stuff. Can we use them? So his idea is to hook it up to, on one side, take an alpha-1,3 sugar, okay, and on one side put a sequence that'll recognize a virus. And then the other side, it's going to be hit by your alpha-gal antibodies, right?
Right.
And so that should neutralize influenza when it gets in you. That's the idea. So the way they link up the alpha-1,3 gal so that it would bind the virus is to make an aptamer. This is very interesting. You could select any DNA sequence so that it will actually bind a protein. You just make a big library of sequences, and among those are some that have the right structure so they'll bind a protein. So you just
there are ways to amplify, screen and amplify these libraries until you get out the aptamer that you want. We've got a guy here that does this, and it is very cool.
It's neat. So you put the aptamer on the alpha-1,3 gal. I guess you put it in yourself when you get influenza, and it would neutralize you, right?
And the idea of doing this, his idea of doing this for HIV, I don't think that's crazy at all. I think and he brings up the idea that if HIV came up with variants that they might be less fit. This is something we've talked about before. HIV is very clever at coming up with variants that are just as fit. So you're stuck with some of the same sorts of problems that people have in trying to make vaccines. I think that you'd have to try and make aptamers that reacted with conserved bits of the HIV surface and et cetera. Nevertheless, in a general sense, the idea is perfectly reasonable.
Yeah, it's not a bad idea. I think for HIV it makes sense because there you have a easily diagnosed long-term infection that you can treat harder with flu.
Right.
But the problem with HIV, though, is late in infection when you're not making much of an immune response. You might not have many of these antibodies left, right?
Oh, that's interesting, yeah.
So, but I think it might be something we should look at. Certainly do some clinical studies. Hey, it's a good idea, Eric. Sure. Thanks for sending that. Thanks for sending the voicemail, too. Our next email is from Joseph. Dear TWiV, I'm a graduate student in the Computer Science Department, University of New Orleans, studying bioinformatics. I only recently discovered that biology is nothing like what was presented to me in high school, and have found this an excellent service to fill those knowledge gaps I have naively created. I am emailing a response and a request. It's an interesting way to look at this, Rich. It's not like high school biology, huh?
Well, unfortunately, high school biology lots of times is lists of things that you're supposed to remember, rather than teaching, how we know stuff and asking a lot of questions and trying to look into the logic of things. All too often, it's just a bunch of memorization, and that's too bad because high school's an important selection for who's going to go on and be interested. Yeah. Teachers are everything. When I interview students for applying for either graduate school or medical school, quite often what I'll ask them was, "What was your favorite course? What was your least favorite course, and why?" And invariably, the favorite course was the favorite course because of the teacher.
Yeah.
And the least favorite course was the least favorite course because of the teacher. It doesn't have anything to do with the subject matter. And that's why it's particularly important to make K through 12 education a respectable respectable career. So that and pay those people really well, so that we can put good people in and educate students, capture their imaginations when they're young.
Now, you ask a scientist what inspired you to go into science. Most of the time, a teacher in high school.
A teacher, exactly.
So, I'm looking at my son's freshman high school biology. A lot of good stuff, really good stuff, but he doesn't care, and the reason is because his teacher is not inspired.
Right.
But if you had someone that were inspiring, they would all love it, or many of them would love it. And that's the difference between a really good teacher and just an average teacher, is inspiring. Right. At some point, you requested your listeners provide what other webcasts they listen to, so I created a list of them in 3 orders: favorite, density, and discovered, all from least to greatest. Hopefully this helps. I'm not much of a list person, but I figured it would be more useful than just a list of what I listen to. For your reference, I generally listen while doing non-critical tasks at work, driving commutes to work and campus, and walking, biking commutes to campus.
This person listens to a lot of podcasts.
Yeah, nice list.
Yeah.
So let's see. His favorite is This Week in Google. And he also likes, 1, 2, 3. I have to count in reverse here. 1, 2, 3... 4, 5, 6. Number 7 is Futures in Biotech. 9 is This Week in Parasitism, and 11th is This Week in Virology. Okay. Then we'll put these lists on the website so you can see what else he's listening to.
I'm going to have to listen to This Week in Google. Wonder what that. Have you ever listened to that?
Yeah, I actually like it. It's about cloud computing, Google-related things.
Okay.
Computing in the cloud. It's This Week in Tech Network, Leo Laporte, and Actually, one of the guests is a professor at City College here in New York City.
Right.
He's a journalism professor. So then we have a list according to density. The density of the podcast, I guess, right?
I guess.
It's pretty much the same order. This week in Google, Futures in Biotech, this week in Parasitism, this week in Virology have more or less the same ranking. And then Discovered. Recently discovered? I don't know.
Maybe.
Oh, okay. So the most recently is Berkeley Speakers, and then we have This Week in Parasitism, This Week in Google, and This Week in Virology.
That would make sense, yeah.
Okay. And there are a bunch of other interesting ones which listeners might want to see, so we'll post that.
That's a nice list.
And then he goes on, with that aside, I have a question request. I'm new to the biology field, and I'm working with a bioinformaticist this summer. Specifically, we are collaborating with some biologists who have information which they published: human T-cell leukemia virus type 1 integration, target sites in the human genome, comparison with those of other retroviruses. That was a Journal of Virology paper. On preferred integration sites of HTLV-1 in HeLa cells. This is contrasted with other virus families' preferred integration sites. Specifically, our goal is to create a model from the HTLV preferred integration sites, hopefully find other studies using HTLV-1 with different cell lines, and incorporate that information into a model which will predict integration sites of HTLV in a T-cell line. Naturally, the ultimate goal is to have a general enough model so that given a virus's phylogeny, we can predict its preferred integration sites. Do you know of any similar studies with HTLV integration sites? And as a complete amateur, what materials tools would you recommend I review? I'm currently familiarizing myself with UCSC Genome Browser and tackling any vocabulary issues I have in the aforementioned paper. Thanks, I love the show. Maybe shoot for more metaphors when explaining processes or comparing, contrasting things. Joseph.
All right.
Yeah, Rich is good with the metaphors.
Yeah, I'll work on it.
P.S. I encourage TWiP on a bi-weekly basis, but I have a lot of TWiV to catch up on, so no rush. Well, I suggest you go to PubMed. I don't know if you know about that, but...
That was my thought exactly.
Do a search, HTLV-1 and integration. Put and in between your keywords. I found a couple of papers right away, so there are some other studies on it. And I think that's the best place to look. That's where scientists look for the literature, right?
I did a search just to broaden it up a little bit and put retrovirus integration site selection. Came up with 130 hits. Some of those articles might be quite interesting because I wouldn't look at just HTLV-1. I would sort of broaden my interest to include other retroviruses as well, because that could inform you about HTLV-1. And keep in mind, some of these papers that you found, Vince, they're looking at people with disease.
Mm-hmm.
So when you're looking at somebody who's got a disease, if the integration site has anything to do with the disease, then you're selecting out of the total possible, a total population of possible integrations, the ones that cause disease. So you have to make that sort of differentiation as well.
Yeah, that's a very good point. Yep. So it becomes clonal in that person, and so you want to look in cell culture too. But probably among those that you found, there are some which are studied in cell culture.
Right. Okay.
There you go, Joseph. Okay, we have an email from Jose. My name is Jose. I'm a PhD student in Mexico City, and I'm working with innate immunity against dengue virus infection. Well, I hope you liked today's discussion, Jose.
Yeah.
First of all, thanks for the great job with the cast. Before I became a fan of TWiV, I knew your work with RNA detection and innate immunity, and I never thought that doctors of your level were so enthusiastic with the academic part of science, making it so fun and interesting.
You know, when I started out, I thought I wanted to be a teacher.
Is that right?
And that research was sort of what you had to do to get a teaching position in a university like I was at. It took me a long time to figure out that it was the research that really kept me going. Teaching as well, but it's a combination of the two.
That's interesting. Well, I didn't think about teaching for many years. I wanted to do research, and then I taught. It was something we had to do, and I really liked it.
Right.
So it got to be more and more of what I like to do, and now we do this. So not everyone is enthusiastic, Jose, about teaching. I'll tell you.
That's true.
Some
That's
people
true.
don't want to teach.
And it shows.
And if they have to teach a lecture, they'll just give a research seminar, and it's not the same thing.
Right.
I get complaints about that here all the time. All right, back to Jose. I have some comments and questions. One, are there any viruses we know that are transmitted in seawater? Since there are so many of them in my sea, my guess is that some of them are capable to infect humans. Do you think that some of these viruses actually use the seawater for spread? Might be very interesting. Evaluate the stability of the particles since they are resistant to high concentrations of salt. Yeah, absolutely. So I don't know a lot about this, but I know that caliciviruses infect many marine mammals.
Okay.
Whales,
Good.
seals, dolphins. Whales excrete 10 to the 13th particles a day in their feces.
Holy cow. Where'd you come up with that
Sorry,
number?
per gram of feces.
And a whale probably produces quite a few grams of feces.
Yes. So,
Wow.
and these viruses, they don't really harm the whale, but guess what? They infect people. They can cause rashes, gastroenteritis, fevers, and they often cause outbreaks. And here's something I learned recently, which is very interesting. You go to one of those water shows where you watch whales jumping out of the water.
Right.
They're spraying you with viruses.
Oh, my goodness.
Outbreaks, these calicivirus outbreaks often happen in water parks.
I'll be darned.
Isn't that cool?
That is cool.
And there are often outbreaks on shore regions, in waters, calm waters where marine mammals are calving, giving birth, right?
Mm-hmm.
So the high concentration of virus there, people swim and they get infections. Very little known about these, but I find them really fascinating.
That's very interesting.
Because you're right, there's the high salt. They must be really stable, or maybe the animals make so much of them, they just get around that. But and then, of course, once people get them, they spread them amongst themselves as well. So there you go. That's one idea. All right, his second question. There are many reports of exclusive detection of viral PAMPs by toll-like receptors and rig-eye-like receptors. Appear to me like the mechanism of detection depends on the cell type, the genotype of the virus, the genetic composition of the host, not just by the pathogen recognition molecule involved. Since almost all of these kinds of receptors activate interferon regulatory factors and the NF-kappa B families of transcription factors, these differences in the specific combination of elements in the detection part can give rise to the activation of a specific dimer in the case of NF-kappa B, and for interferon regulatory factors, given to an activation of different sets of genes in each case by differences in the affinity for the promoter sequences and the quantity of transcription proteins. So the response, it's more complicated than just activated the IRF family and NF-kappa B pathway.
Absolutely.
Yeah. Did we do that? Probably.
Yeah.
Yeah, well, sometimes we can't really get into the details, right?
Right.
But you're right.
But he's absolutely right. It's very complicated.
Nothing is really, really simple, but we try. So the audience of TWiV is broad enough that we have to appeal to people, which isn't bad because even for me, I like simplifying. But anyway, you're right. 3, about the T-cell specific retroviral therapy question in TWiV 71. So that question was, is it possible to use gene therapy to insert genes into the HLA complex that could encode protein expressed on T cells for a certain virus? So this is just what we were talking about.
Right.
Right? And then he wanted to know if you could actually put the peptide in the MHC complex.
That's right. Yes, I remember that.
So he says one of the obstacles is that the region where the specific epitope is loaded in the MHC, which is the same as HLA, is not linear in sequence. It's formed by the spatial structure of the MHC. So if a virus has to integrate that sequence that codes for the specific peptide, it must do it in fragments that have to fit exactly in the gaps of the MHC and has to leave the sequence in the open reading frame. I think that's very difficult.
That's actually a really nice answer. That's very good.
And then finally, he sent us a paper, said, look, he's very excited about this. Look at these microscopy techniques that allows visualization in the range of 40 nanometers. So cool. Thank you for the great show. And sorry, my English is very precarious. That's fine. It's fine, Jose.
Communicates just fine.
So the name of this paper is Dynamic Cluster Distribution of Hemagglutinin Resolved at 40 nanometers in Living Cell Membranes. So it's pretty cool. They have nice images in this, and they use basically a laser-based microscopy technique. It's called fluorescence photoactivation localization microscopy, and they can resolve 40 nanometer structures basically, or at the resolution of 40 nanometers which is sublight, but can be done with lasers.
Right.
And they look at this HA and they can see that it has some interesting characteristics in the membrane that don't fit with a lot of the membrane theories, right?
Right.
I think
nice little movies. Yeah.
Nice movies, very cool. It's a PNS paper. We'll put the link for that. Thanks for sending that. And I think that we will stop our email there and move on to picks of the week so we can finish in a good amount of time.
All righty.
Is that okay?
Sure. So my pick is a book that my son-in-law and daughter gave to me for Christmas a while ago. I think they just said, Dad's a science geek, and so this looks like a science book, and they just kind of picked it up, but they really hit the bullseye. It's called The Mold in Dr. Florey's Coat, the story of the Penicillin miracle.
Nice.
By the author is Eric Lax. So the deal is that everybody, when they think of penicillin, think of Alexander Fleming, who discovered that the penicillin mold made something that would kill bacteria. But it might not have gone very far from there had it not been for this gentleman, Florey. I'm blanking on it. It's Howard, right? Yes, Howard Florey was Australian to start with, but did had his career in England. Mm-hmm. Who in the 30s and 40s took on the task of, he read Fleming's paper and said, this looks interesting. We need to purify the active ingredient and see if it can be used. And it's a story of how they did that. And it's really well written and really an amazing story. So
nice. Yeah, it's kids don't know what to get you after a while, so
I recommend that. And there's actually I looked up the Wikipedia site on Florey just for kicks, and there's a couple of interesting quotes. This one I like. He said he won the Nobel Prize for this work, shared it with Fleming and one of his co-workers, and he says, people sometimes think that I and others worked on penicillin because we were interested in suffering humanity. I don't think it ever crossed our minds about suffering humanity. This was an interesting scientific exercise, and because it was of some use in medicine, is very gratifying. But that is not the reason we started working on it. Isn't that interesting?
I think that's probably very typical.
Yeah, very typical. Just a scientific challenge, and it's important to keep that in mind because a lot of, there's a lot of pressure for people to work on stuff that is really important in medicine, and I appreciate that. But a lot of the most important discoveries that really impact on medicine are not made because they were motivated in that way. They're made because people were just interested, just curious.
No, it's a great point. We just had a seminar by Marty Chalfie, who won a Nobel Prize recently for producing green fluorescent protein in C. elegans.
Right.
And his whole theme was, this had no application whatsoever, this idea of ours to put this protein in worms. And he said, that's the way research has to be. Don't make directed research so crucial. Let people just fool around, because now this is incredibly important in all fields, right? So don't tell us that we have to work on a disease, no translational emphasis. Just let scientists wander and good things will happen.
There's another bit from this wiki article on Florey that I liked that we actually wound up talking about today. Maybe it was in the back of my mind. It says he was openly concerned about the population explosion resulting from improving healthcare. And he's a staunch believer in contraception.
Interesting.
So, that's an interesting thing to think about. Penicillin has had a profound effect on healthcare. It's probably one of the most important discoveries out there.
Yeah.
And probably increase the average lifespan by a significant amount. So,
I wonder what the effect of that will be. You know, Dixon is always talking about how parasites are really important for keeping life forms in balance.
mm-hmm.
So we're removing all that. Check.
Right.
And,
That's right.
you know, with antibiotics, antivirals, vaccines, drugs of all sorts. So
Following up on last week, I was reading about allergy.
Mm-hmm.
And there's a lot of theory about how what we experience as allergy was originally an immune response to parasites. And since the parasites aren't around anymore, it goes wonky.
Right.
And there's also a lot of theory. There's the hygiene hypothesis behind allergy, that if we are too hygienic,
that's right,
that we wind up tinkering inadvertently with our immune systems and wind up with more asthma, more allergies, et cetera. And apparently that theory has a fair amount of backing.
Yeah. So there you go. Who knows in 50 or 100 years what will be the outcome of us fooling with this, you know?
Right.
Great. That's very nice. I put that on my list when it comes out for the iPad or what's the other one? Kindle. Okay. So my pick was suggested by Eric, who is the author of the voicemail that we read. And it's a site called Proteopedia.
This is amazing.
It's a very nice site. It's a collaborative 3D encyclopedia of proteins and other molecules. So you go to the front page, and there's a little Java that has to load. But when it does, you see today, at least, there is the ribosome. It's the featured molecule.
And I'm so glad. I don't know if the ribosome comes up every time, if that's the featured molecule all the time. But I really feel lucky that it was here this time, because ribosome is one of my favorite guys, and the ability to toggle all these different parts of it on and off, it's just amazing.
It's great. So in this picture, they have the three sites, the PA and the E sites for the tRNAs. You can see them right there. Gives you a really nice feel for how this whole thing is put together.
It's amazing.
Beautiful. So lots of good molecules, three-dimensional structures. You can see pictures, rotate them. It's very cool. So
it's got another thing down here.
Yeah.
Down on what's new. I don't know how long this will last, but there's a link to a mini biography of Frederick Richards that caught my attention, because in fact Fred Richards was one of my teachers in graduate school.
Yeah, how cool is that?
He is he fairly recently passed away, a world-class crystallographer. And it's got a picture here on the biography of him, which is really nice, of what they call, has two names, either the Richards box or Fred's Folly, which is this thing that he invented to put together protein structures, build models from crystallographic data. It's arranged so that you can build a ball and stick model that's like huge, like 4 feet in diameter. Inside this box, you can walk around and tweak the little balls and sticks on this huge model, while superimposing on it the individual planes of density from an electron density map. That was before computers. The first way it was done. He had one of these. I toured his lab as a beginning graduate student and walked around in this thing, and I didn't realize how special it was at the time.
That's cool.
Really
so you were over there at Oxford?
No, this was at Yale. He was ultimately the chair of the molecular biophysics and biochemistry at Yale. Was there for a long time.
He was obviously at Oxford at some point. And this photo was taken there. Yeah, that's neat. I can see where that would be useful. Yeah, that's great. Nice site. Thanks for that, Eric. So check it out. On the front page, by the way, if you have a mouse with a scroll wheel and you put the pointer over the structure and scroll it, you zoom in and out. And you can go all the way in to see individual molecules. It's a really cool site. It's like a wiki-type site.
It's very cool.
Very nice. And that will do it for another educational TWiV. Can we call it that? I guess so.
Sure, why not?
As always, if you're new to TWiV, please go over to iTunes, subscribe. It's free, and you automatically get each new episode, and post a comment. say something nice if you feel so, and that keeps us on the front page of iTunes. Tell your friends about it and spread the word about TWiV. We also have begun to work with Stitcher, which is a way to listen to TWiV on your mobile device, your mobile device, your iPhone, your BlackBerry, Palm, Google Android. It streams TWiV to your device without having to download it. You can go to bit.ly/StitchTWiV, all one word. They have a landing page for us there. It works pretty well. I was trying it out the other day on an iPod Touch. You can subscribe to TWiV, and it tells you when there's a new one. You just click it, and it plays it.
I tried it on my iPhone. Works just fine.
It's free. Just another way we can get you virology information wherever you are in the world. TWiV's part of sciencepodcasters.org, promednetwork.com, and microbeworld.org, where you can find other science podcasts and a whole lot of other stuff. And next week, we're going to have a special guest on TWiV. We're going to have Dr. Kirsten Sanford, the host of This Week in Science, TWIS, and Dr. Kiki's Science Hour, which is on the TWiT network. So that will be fun. We'll talk to her about how she went from a PhD to doing science education on the internet. And then she'll stick around and talk about viruses with us, so that'll be fun.
Great. I look forward to that.
Stay tuned. As always, send us your questions and comments to twiv@twiv.tv. We love to get them. And next week, besides Dr. Sanford, we should have Dr. Despommier and Dr. Dove. We'll have a big crew. That'll be fun.
A full boat. That's great.
That'll be fun. Hey, Rich, good to hear you again.
Great to be here, as always.
Always fun. And next weekend, I'll be seeing you in person.
That'll be good fun. Well, I'm working on the weather for you.
Just make sure it's humid and hot.
I'm going to get it stinking hot.
And you know what happens when it's hot and humid? You need to quench your thirst. Get rid of those herpes viruses.
We'll take care of that, too.
All right.
Good enough.
You've been listening to This Week in Virology. Thanks for joining us, everyone. We'll be back next week. Another TWiV is viral.